All right, so we're going to do the last learning target for your Section 1 test in Unit 3, which is going to be cellular growth and the cell cycle. So you'll probably need to pause this video at different points to be sure you get especially the visual and um, word information for each of these cell phases in the cell cycle. It's going to be extremely important moving forward if you don't already have that. So organism growth. So growth is an increase in size of an organism. And as an organism grows, the number of cells will also increase. And this is again this kind of how things operate, and it makes sense as things get bigger, then the things that make up those things need to increase as well. And then we're talking about cells in this certain situation. So some limitations of cell size. The first one is DNA overload. So we do know, and by this point, that each cell has a copy of DNA, and that is responsible for the production of proteins. So if the cell gets too large, then the DNA is unable to produce or code for enough proteins to keep the cell functioning properly. So this limitation limits the size or how large cells can get. And with that being the case, then it plays a role into why cells divide. Okay, if cells didn't have to divide, then you would have one cell that just continually grew bigger and bigger and larger and larger. But unfortunately, there's only so much proteins that can be produced off of the blueprint, which is the DNA. And remember that proteins give life, give way to life. So if you don't produce enough proteins, then you can't carry out the functions that are needed within the cell. Another limitation is material exchange, excuse me. So as the volume of the cell increases, the surface area of the cell does not increase as fast. Therefore, it limits the amount of area for the cell to not only take in essential materials from the outside, but also to eliminate waste from I should say inside, not outside, excuse me. So cells with higher surface area to volume ratio are more efficient. So as the inside of the cell grows, the outside of the cell may not grow as much, which means that there's not going to be able to be an increase of materials moving in or out of the cell, which is going to limit how large the cell can become. So the definition of surface area, this is the amount of space the outside of the cell takes up. And volume is the amount of space taken up by the entire cell. So surface area to volume ratio, high surface area to volume ratio equals a more efficient cell. That's important to know. In order to maintain a particular surface area to volume ratio, cells will divide. Just like most things that are living, there's this ideal area that things operate the best, and cells are no different. So there's an ideal surface area to volume ratio where cells are as efficient as they are going to be, and that's where they like to stay. All right, so the cell cycle. So this is the life cycle of a cell, and the life cycle of the cell ends with the death or the division of the cell. Okay, so once a cell dies, of course, the life cycle is over, or once a cell divides, then that particular life cycle of the cell is over and a new one begins. Chromosomes, we've talked about this before. The chromatin of the cell is condensed into chromosomes. And once we enter the M phase, then this is when this occurs. Okay, the chromatin will condense into chromosomes, then it will wrap around proteins called histones, which gives them the X shape here. That are the sister chromatids that most of us think about when we think about chromosomes. So chromosomes, when chromatins condense, it winds around proteins called histones. The histone plus DNA unit is called a nucleosome. Nucleosomes group together to form chromosomes. The centromere is a point of attachment during mitosis. This is usually near the center of the chromosome. Sister chromatids are after the DNA is replicated. This is two copies of chromosomes that are attached at the centromere. So the phases of the cell cycle. You'll have the interphase, Then you have mitosis, or also called the M phase, and then you'll have cytokinesis. So if we look at this pie chart over here, notice that the cell spends a majority of its time in interphase. Okay, M phase is a very small portion of the life cycle of the cell, with cytokinesis being even smaller. So that's important to know. Okay, most cells spend a majority of their time in interphase. Now, during interphase, it may be prepping for M phase, which we'll talk about, but a large chunk of the cell's life cycle is spent in uh, interphase. So, talking about interphase, this is the phase between cell divisions. This is when normal life of the cell takes place. An interphase is broken up into three separate phases. So you have G1, S, and G2 phase. And each of those phases plays an important role. And some of it is just the cell living its normal life. Some of it is when the cell is going to be preparing for um, the M phase. So the G1 phase, and this is the first growth phase. So in the G1 phase, the cell is coming out of cytokinesis. Okay, so you have a brand new life cycle that is occurring. We've completed the M phase, and we have completed cytokinesis, and so now you have a new daughter cell. So this is when the cell will grow, organelles are replicated, and proteins are synthesized. During the G1 phase, the cell is going to go about its normal life, functioning of what it's made to do and doing what it's supposed to do. Okay. Then you move into the S phase, and this is the synthesis phase. DNA is replicated during this phase. So, for example, humans have 46 chromosomes. After the S phase, they will have 92. When the cell finishes this phase, it is usually going to divide. And the reason being is because of the amount of chromosomes. Has to divide. Then we enter the last phase of interphase, which is the G2 phase, and this is the secondary growth phase, and this is also the preparation for entering into the M phase. So centrioles are formed, and the cells finish preparation for division. When the cells finish this phase, the cells will enter into the M phase. So then we enter into the M phase. Okay, this is when the cell undergoes a division of the nucleus and all the organelles. Mitosis is broken up into four separate phases: prophase, metaphase, anaphase, and telophase. So prophase, it's important to be able to visually see what's happening in prophase as well as being able to describe what's happening with words. Chromatins condense into chromosomes. The centrioles separate and move towards the poles, okay, opposite ends of the cell, and spindle fibers form as the nucleus envelope begins to break down. So you'll see the centrioles here begin to move to other ends of the cell. If you look up here at a dyed live color of a cell, these green look like starlights or laser beams are the centrioles. Okay, the nuclear envelope begins to break down. Okay, and then spindle fibers, which are a form of cytoskeleton we talked about, begin to form as well. So if you want to pause this video, draw this in your notes, I would suggest it. Then you have metaphase, and the big thing that happens in metaphase is the chromosomes line up across the equator, or the center of the cell. Okay, the centrioles are at opposite ends of the poles. The spindle fibers have attached to the centromeres, okay, which are the center points of the sister chromatids, and the chromosomes line up across the middle of the cell. Okay, so if we look at this picture, it's not as neat and organized as this drawing, but you could still see that is what's happening. Anaphase. Sister chromatids will separate into individual chromosomes and move toward the centromeres. So the spindle fibers are going to act as tow ropes and pull the chromosomes to opposite ends of the cell towards the centromeres that are at the poles. So if we look here at the live picture, we're beginning to see that happen. We can see a separation happening across the center of the cell. Telophase is next. Chromosomes gather at the poles and begin unraveling with two new nuclear envelopes beginning to form. The cleavage furrow forms, and what the cleavage furrow is where the two new cells begin to pinch, away from each other. And we can also see this here. We see an indentation happening, what we would have considered it during the M phase as the center or the equator of the cell. All right, so M phase is over. We move into cytokinesis, and this is the actual splitting of the parent cell into two identical daughter cells, also known as the act of the parent cell becoming two new identical daughter cells, animal cells. The cleavage furrow completes, and the cell pinches forming two new cells. And so the cleavage furrow will pinch on, off, and break off in animal cells. In plant cells, a cell plate is going to form in between the two new cells. And the reason this is because of the cell wall. Okay, the cell wall is not going to pinch in two. Remember, it's a harder structure that gives support and stability to plants. So a cell plate is going to be formed in between the two new cells in plant cells. And that is important to remember. So cell regulation, you have these things called checkpoints. Okay, so there's a series of checkpoints in the cell cycle that ensures the cell is completing the cycle without any problems. And this is important because you're copying and reproducing genetic material, which is the blueprints for all of the proteins that are going to be made. So if an issue happens, especially with the genetic material, then we could see some very, very major consequences arise in cells. So one way cell cycle is regulated is contact inhibition. And this is when cells are touching other cells. If this is the case, they will stop growing because there's no room left for them to grow. The picture is a petri dish. You could, culture cells in a petri dish on a certain type of augers and in a autoclave. Those cells will replicate and grow until the entire petri dish is full of cells, and then they'll stop because there's nowhere else to grow, there's nowhere else to go, so they're not going to continue growing on top of each other. Then you have cyclins, and these are proteins that control the cell cycle. They are produced to initiate mitosis and cease the production during mitosis. All right, so what cancer is an irregular growth and division of cells. And this is when there is a breakdown in the regulation of the cell cycle. This causes an abnormal growth of cells called a tumor. So, for instance, contact inhibition will not stop cells from growing. They will just continue to grow on top of each other and continue to grow and continue to grow and continue to grow, even when they're not supposed to. So, any type of breakdown in the regulation of this cell cycle is a cancer. And then cancer is usually caused by mutation of the DNA, which causes a faulty protein to be produced. Mutations on the DNA can be increased by substances called carcinogens. So, as examples of carcinogens is cigarette smoke, pesticides, burned food, anything that can increase the chances of mutations or alterations to the DNA of cells. And so the last thing I'll leave you with is how you kind of remember the cell process. And I should have said this during the phases of the cell, but how I remember the cell process in order. Okay, IP-MAP, interphase, prophase, metaphase, anaphase, telophase, and of course cytokinesis at the end. IP-MAP puts them in order. Now you'll need to remember that in interphase you'll have the G1 phase, the S phase, the G2 phase. But just being able to write down those letters, IP-MAP, in a row or in order, will be able to, or should be able to, help you remember the cell cycle. Be sure you complete the video form. If you have questions, let me know, and we'll prepare for our test. Thank you.